Hi everyone. So I'm back with a question and a discussion on the paper 3 experiments, as many of you will be taking the SPM practical paper 3 coming up soon, right? So this is SPP 2023 paper 3 about diffusion across a Visking tubing. Okay, and here we are going to discuss how the size of the molecules affect the process of diffusion across the Visking tubing. Okay, so in this experiment, you will also receive some tips on how to write a procedure. So this question is different from the other questions that I've put up so far because it will help you to prepare yourself in case you have to write a procedure. Okay, there are two types of questions, right, in the biology paper 3. It can either be that the procedure is given to you and you just carry out the procedure for the experiment, so that's easier. But a more challenging question will be they ask you to write the procedure based on the aim of the experiment and maybe a picture of the experimental setup. Okay, so how do you write procedure to get your maximum marks? Usually it's 3 marks. How do you get the maximum 3 marks? What are the marks based on? Okay, so we're going to discuss this, and also there are many other tips to help you to prepare you to answer the paper 3 question that's coming up in your exams. So take this as a guideline, all right, or some strategies to answer a paper 3 question. Not necessarily the same question here will come out in the exam, right? All right. So be prepared for any type of questions, and to be prepared, you learn the techniques of answering instead of just memorizing a few experiments, right? Okay, so this paper is very rich in all the tips and the last minute pointers to help you score in the paper 3. So do go through it. Now, dream big, stay focused on your revision and your dreams, and believe in yourself no matter what. Never give up. So let's begin. Now, the first page of the experiment is to check your apparatus and materials. So you're not allowed to work with the apparatus in the first 5 minutes. This period is used to check the apparatus list, read the question, and plan the experiment which will be carried out. And mark tick in the box provided to check the material and apparatus provided. Okay, so let's look through the apparatus. All right, imagine you are taking the exam now. Okay, and let's look at it. So first, there's a beaker containing solution Q. It looks like this. Now, can you guess what this item is? What solution is it? It has a yellowish brown color. Yes, this is actually iodine, as you'll find out. Okay, and then there's a beaker containing solution P, which is like a cloudy suspension, whitish in color. Okay, if iodine is involved and there's only one other solution, what can this be? Yes, this must be starch. All right, because iodine is used to test for the presence of starch. You will find out later as you do the experiment. Now, then there's a beaker containing distilled water. There's a Visking tubing, which is around 15 cm in length, a thread, then a digital or analog stopwatch. Here is shown an analog stopwatch, which is the old type of stopwatch. So you're going to time 15 minutes for the Visking tubing to be immersed in the solution. All right, it's 15 minutes. Then syringe without needle, okay, as shown here. This is to help you draw out the solution P and put the solution P in a Visking tubing, which is tied at the bottom here. All right, so it's used to draw out the solution P from the beaker into the Visking tubing. Then distilled water and a small towel. Right, so here are some tips on how you can get started. Firstly, let's look at the question. You are required to carry out an experiment to study the movement of substances across a selectively permeable membrane based on the different sizes of molecules. And the diagram 1 shows the setup of the experiment. Okay, so as you can see here, there is a Visking tubing, and this is actually the selectively permeable membrane, right? And then you have two solutions, solution P inside the Visking tubing and solution Q with the distilled water outside here. All right, so you are not told how many ml or cm cube of the solution P and Q to use. You have to determine it yourself. Okay, now the first tip is actually to find out the objective. So where is the objective or aim? It starts with the word to study or to investigate something. Okay, so starts here, or to determine something, to determine, study, or investigate something. All right, so find the objective. The objective is the movement to study the movement of substances across a selectively permeable membrane based on the different sizes of molecules. Then from this objective, determine your main MV and RV. Now, these are my own personal terms. Main means that it is the MV or RV from the main aim or objective. Okay? And the sub MV and RV are the operational MV and RV. These are the ones that you use in the lab, you handle in the lab. Okay, the sub RV is the data that you collect, the observations or the measurements you make, not the calculations yet, the observations and measurements that you make in the lab. Okay, the raw data that you collect. So, okay, what is your main MV and RV from this statement? Now, you can see that the experiments to study the movement of substances across a selectively permeable membrane, that means the Visking tubing, based on the different sizes of molecules. Different sizes. The word different here hints to you that this is the MV. Okay, it's based on that. So when you change the size of the molecule, then the movement of substances across the Visking tubing will change, whether it can move through or cannot move through. Okay, so that means your MV now, the tick 2 is to draw a table of variables. I call it the TOV, table of variables. So what is the table of variables? Now, it is a table consisting of 3 columns with the word main and sub here, and then 4 rows, okay, with MV, RV, and FV. So all this you can, if you have a rough piece of paper, you can ask them for some pieces of paper where you can jot down these things, all right, or you can just sketch it at somewhere at the side. So in this table, I'll use a pencil to draw it so you can correct it if it's wrong. Write down your main MV from the objective, which is the size of molecules, right? In this case, the molecules come from solution P and Q. So we call them molecule P and molecule Q, right? And then there is no sub-MV in this case because it remains, this MV is the MV for the experiment too, okay? It's the main MV is the MV for the experiment too. Now, how about RV? No, so for RV, let's look at this one. To study the movement of substances across the Visking tubing, right? So the RV is to determine whether the molecule can move through or cannot move through the Visking tubing, right? So RV is the ability of the substance or the molecule P and Q to move across a selectively permeable membrane, which is your Visking tubing. Okay. By the way, these 3 variables are taken from the textbook too, so it's confirmed correct. All right. And then the FV is the temperature as stated in the experiment in chapter 3. All right. Temperature. Okay. But the experiment in chapter 3 is a bit different from this one. Okay. I'll tell you later. So sub. Now the sub MV, there's no sub MV. The main MV is retained. Okay, in the experiment. Now, how about the sub-RV? So sub-RV is the data that you collect or whatever you measure in the experiment. So to find out the ability of molecule P and Q to move across the Visking tubing, we have to detect the presence of molecules P and Q in the Visking tubing and beaker. Okay? Now, how are we going to detect the presence of these molecules? You have to detect it by detecting by using a test. In this case, the iodine test is used. Okay, so the presence of the molecules P and Q are detected by the change in color of the solutions in Visking tubing and beaker. Okay, by using the iodine test. So this is quite an ingenious experiment because we have placed iodine in the distilled water and starch inside the Visking tubing. So if starch managed to diffuse through into the water here, into the iodine solution will turn blue-black. And if iodine can diffuse through or move through the Visking tubing, then the starch suspension in the Visking tubing will turn blue-black. You see, so it's quite an ingenious type of experiment. Okay, quite interesting. However, in your textbook, glucose is used. Glucose is also put into the Visking tubing together with starch. But here, there's no glucose involved. Okay, if glucose is involved, you have to test the solution outside for glucose by using Benedict's solution. Okay, but in this case, there's no glucose involved. So only the iodine test is involved. All right. Now, let's come back to our question. So after you have done number 1 here, tick 1, and tick 2, you have drawn this out, you're clear about all this, then you know it's very easy to uh, carry out the experiment and plan the procedure. Okay, so plan this experiment by using the apparatus and materials provided. Your procedure should include method to handle the MV, method to handle the RV, and precautionary steps. Okay, so these 3 will get you 3 marks. So you must mention how you handle the MV, the RV, and the precaution. 3 marks. Okay, so for every experiment, you have to find out how to handle MV, RV, and what are the precautionary steps. 1 precautionary step is enough. Okay, but if you're not sure, you write in a few. Okay, so 1 mark for each here, each item here. And by the way, the hypothesis as from the textbook is only small molecules are able to diffuse through a selectively permeable membrane like the Visking tubing, but larger molecules like starch cannot diffuse through the selectively permeable membrane. So what's the small molecule used here? Yes, it's the iodide ion or the iodine molecules, right? That are present in the iodine solution. All right. So this hypothesis is also taken from the textbook. Now let's go on to the procedure. All right. So as I said now, the tip is that for every experiment you study, please memorize the how to handle the MV, RV, and FV and the precautions also. Okay. Now in this case, the procedure, you have to write from number 1 to number 12 to write the whole procedure. But make sure to include your MV, RV, and the precautions. Okay. So let's go through it. Number 1, prepare a Visking tubing of length 15 cm. How to prepare? Soak the tubing in water for 5 minutes first to soften it, or for a few minutes. Okay, but not too long. So your time is quite fixed, so make sure that you are conscious about the time that you have, which is about 40 minutes to carry out the experiment and answer the questions, right? Now, next, tie one end of the Visking tubing with a thread tightly to prevent leaking. So this adjective is very important, tightly. That is your precaution. So tie one end of the Visking tubing below here, tightly to prevent leaking. And then fill the Visking tubing by using a syringe. You fill the Visking tubing with 15 ml of solution P. Okay, that is the starch suspension actually. Okay, so fill the Visking tubing with 15 ml. Now the volume you have to determine yourself, right? Then tie the other end of the Visking tubing tightly with the thread here. Again, tightly. You must have this word to so that is a precaution. Tie the other end tightly. Okay. So there are 2 precautions here, and there's one more. Rinse the outer surface of the Visking tubing with distilled water to remove any traces of starch, okay, that might have dripped onto the outer surface of the Visking tubing. So this is another precaution. So any one of these precautions will be able to get you that 1 mark of precaution. But you just write down everything, right? The 3 precautions. Next, put or pour 500 ml of distilled water into a beaker. All right. So 500 ml of distilled water, put it into the big beaker, and then add 10 ml of solution Q into the distilled water. That's the iodine solution, right? So this is also part of MV because you have to state what you do with the solution P and what you do with the solution Q, because these 2 have molecules of different sizes, right? That is the starch molecules and the iodine molecules or iodide ions. Now next, immerse the Visking tubing in the distilled water and with a Q, distilled water plus Q, right? In the beaker. So immerse the Visking tubing in the beaker like this. That's for 15 minutes actually, right? Now 9, record the initial color of the solutions inside the Visking tubing and outside the Visking tubing. That's at the beginning, right? So when you look at these 2 solutions, solution P will be a cloudy white solution, white cloudy solution, right? And solution Q plus distilled water, what color is it? Now you have to be very careful. Don't mention, when you record, don't mention it as a yellow color. They don't accept. They accept yellowish brown only. All right, yellowish brown. I think it's somewhere in the textbook also, yellowish brown, or in the answers given, right? So yellowish brown solution. Remember this word, yellowish brown solution. That is the color of the iodine solution. Okay, yellowish brown solution outside the Visking tubing in the beaker here. Now that is your RV1. That's the initial color of the solution. Number 10, start the stopwatch and immerse the Visking tubing for 15 minutes. How do you know it's for this time? If you check through the question later on, you'll find that they do mention 15 minutes. So it pays to just flick through the paper and you may get some hints there. All right. So why don't you put it for 10 minutes? Because if shorter than 15 minutes, it may not show the result yet. All right. And if it's too long, you will not have enough time to finish answering the questions in your paper, right? Because you're waiting for the results. So 15 minutes in this case is the time given. So after 15 minutes, record the final colors of the solutions in the whisking tubing and outside the whisking tubing respectively. Okay, now this is the final color, right? So what happens after that? Now we'll discuss. So after the 15 minutes, we'll discuss the result in the next slide. Then record all the observations in the table. Okay, so how are marks given? So marks will be based on your MV, RV, and precaution, like this. So the criteria for the marks. The first mark, M1 or P1, is the method to handle the manipulated variable. Okay, so the examiner will mark here. Okay, P3, you have P3, right? You talk about solution P being put into the whisking tubing. But not only that, you must have P7, add 10 ml of solution Q into the distilled water in the beaker. So if you mention how you handle solution P and Q, then your MV is complete, you get 1 mark. Okay. Now, then the second mark, M2 or P2, is the method to handle the responding variable. Okay. So that is the RV. Now, which of your statements here talk about that? Is P9 and P11, 0.9 and 0.11. Okay. Record the initial color of the solutions in A and B, and then the final colors. Okay. So you need two statements, P9 and P11, to get 1 mark. Okay, when you talk about recording the initial and then the final colors. Now, precautions is easier. If you have written either P2, P4, or P5, you get 1 mark. Okay, so P2 is number 2. Tie one end of the whisking tubing with a thread tightly to prevent leaking. And then P4, tie the other end of the whisking tubing tightly with a thread. And P5, rinse the outer surface of the whisking tubing with this water. Okay, any one of these, you get 1 mark. Okay, so as I said, make sure you know for every experiment what is the precaution, at least one precaution. So 3 marks given here for the 3 criteria, and for MV and RV, there are 2 statements that you have to make to get the full 1 mark, the 1 mark. Okay, so these are the results of the experiment. So in the first, at the initial stage, when you put in the starch suspension P, 50 ml of starch suspension in the whisking tubing, the color is, they just say cloudy. It could be white cloudy solution, right? White and cloudy. And then outside, you have put in some iodine solution into the distilled water, so it will be yellowish brown. Okay, so outside is solution P is yellowish brown. Now, after 15 minutes of immersing the whisking tubing, this is the final result. Okay, the content of the whisking tubing will turn to blue-black color. It's very obvious. So what has happened? Okay, actually the iodide ions or the iodine molecules have diffused from the outside the beaker, outside the whisking tubing, from the solution in the beaker into the whisking tubing, right? They can pass through the membrane of the whisking tubing, which has tiny pores, because the iodide ions are small enough to pass through the pores of the whisking tubing, right? So that's why the iodide ions or the iodine, just call it iodine molecules, will react with the starch and form a blue-black color in the whisking tubing. However, why doesn't the solution outside, the iodine solution outside, turn blue-black? It's because the larger starch molecules inside the whisking tubing cannot diffuse out of the whisking tubing because they cannot pass through the tiny pores of the whisking tubing. So the solution in the beaker remains yellowish brown, which is actually the color of the iodine solution, but the starch did not diffuse out, right? So this is a good experiment to compare the sizes of the molecules of iodine or iodide ions and the molecules of starch. The sizes are different. So let's go on to the next question. Based on the experiment, state the color change that occur in the whisking tubing after 15 minutes. This is when you know that your time for immersing the whisking tubing is 15 minutes. Okay, state the color change that occurs in the whisking tubing after 15 minutes, right? And then explain your observation. So try and answer these questions yourself first, based on what we've discussed just now. Right, let's look at the answers. The solution in the whisking tubing changes color. Okay, you have to state the color change. So it changes color from the initial color is cloudy and then becomes blue-black. Now reject blue. You write cloudy to blue, wrong answer. So the observation is very standardized. Blue-black is the color of the mixture when iodine and starch are mixed together, and this is the color positive test for starch, blue-black color. Okay, so if you write blue also is wrong. You write just black or blue, it's wrong. It's blue-black. Take note of that. Okay. Secondly, explain observation in B1. So here we have to think about the why the solution in the whisking tubing changes color, right? Something has happened, right, to make it change color. So what is the process before that? Yes, it's because the iodine molecules or iodide ions have diffused through the whisking tubing and entered the whisking tubing, then react with the starch, right? So referring back to your hypothesis and aim, you must mention that the molecule of the substance Q, which is found in the solution Q, is small in size, right? That's why these molecules are able to diffuse through the whisking tubing and enter the whisking tubing, okay, where the iodine will react with the starch molecules and then cause it to turn blue-black, correct? Right, so the 2 points are, firstly, substance or molecule Q is small in size, small, must mention it was small, and they're able to pass through the pores of the whisking tubing. Next question, construct a table to record the results of your experiment based on the following criteria. The first one is color of solution in the beaker and in the whisking tubing. All right. Secondly, observation. That means the color of the solution at the beginning of the experiment and end of the experiment. Right. So try and draw the table yourself, but before that, analyze what you should write in the various columns. Right. So first of all, the MV is usually on the left-hand side of your table and the RV is on the right-hand side. All right. So what do you write for MV? Now, color of solution in the beaker. The color is actually the RV, but the solution is the one that's the MV, right? So write solution in the beaker and solution in the whisking tubing. Okay? And then for the observation is actually the color of solution at the beginning of the experiment and end of the experiment. So this word color of solution should come down here. Color of solution at beginning and end of experiment. All right? Now that's the RV. So with that, you can start to draw your table. Okay? Right, try and draw the table and then look at the answers and try to mark your question. See whether you can, mark your answers. Okay, to see how many marks you can obtain. Let's look at the answer. Okay, so the MV is here. The solution in the whisking tubing, which is actually solution P or starch molecules, right? Then solution in the beaker, all right, follow from the first part here, in the beaker that's outside the whisking tubing, that solution Q or iodine solution or iodide and iodine molecules. Okay, iodide ions or iodine molecules. Now, color of solution is your RV. At the beginning of the experiment, okay, follow from here, and end of experiment, right? Then you fill in with the data or your observations. So the solution in the whisking tubing at the beginning is cloudy, is white and cloudy, right? That is the starch suspension. And at the end of the experiment, it turns blue-black, right? When the iodine has diffused into the whisking tubing containing the starch, right? So if you write blue or black, not blue-black, if you just write blue or just write black, it's wrong. Take note of that. Very important, okay? So the observation, the standard observation for the food test, the iodine test is positive result is a blue-black coloration. Blue-black. Okay, remember, blue-black. Don't just write blue or black. Secondly, for the solution in the beaker, which is the iodine solution, at the beginning is yellowish brown. Again, this is a standard term that's used by most marking schemes here. All right. So yellowish brown. and at the end it's still yellowish brown. Now you write yellow or brown, just a yellow solution, yellow, yellow, or brown, brown like that. It's wrong. It's considered wrong. You won't get your marks for the observation. So take note of the standard observation, blue, black for iodine test, and the color of the original color of the iodine solution is yellowish brown. Okay, and it turns blue black when it's mixed with starch molecules. Right. So how is the marking done? Now, MV is 1 mark here. Solution in Visking tubing and solution in the beaker. Okay, and then the RV is the color of the solution at the beginning of the experiment and at the end. Follow from the instructions. That's another mark. Now the third mark is for your data, D1 and D2. All of this considered 1 mark. So altogether is 3 marks. Next, based on the results of this experiment, classify the substances based on their sizes in table 1. Classify the substances. What are the substances that they mean? It's actually the molecule of Q and the molecule of P, or substance Q and substance P. Okay, so the answer, which one has a small size of molecule? Don't mention iodine ions because they refer to the iodine ions as the substance or molecule Q. Okay, so use that. So actually the substance or molecule Q, which is the iodide ions or the iodine molecules, they are small in size and they can diffuse through the Visking tubing. And the large size molecule is P. Okay, don't just write P, you must write the particle, which is the substance or molecule of P. Okay, substance P or molecule P. Don't write solution, don't write solution Q and solution P. It's wrong. It's the particle inside there, right? So it's molecule Q and molecule P, or substance Q and substance P. Now, if you're not sure, you write in bracket substance bracket molecule P, substance bracket molecule, substance bracket molecule Q, substance bracket molecule P. Next question. So this is a prediction question. A pupil repeats this experiment by placing solution P with juice X into the Visking tubing. Now let's look at this diagram. It shows a farmer harvesting the sugar cane, and then after that producing the juice X, which is actually the sugar cane juice. Okay, and then let's look at the question. A pupil repeats this experiment by replacing P, solution P, with juice X in the Visking tubing, right? Now predict the color changes in the Visking tubing after 15 minutes, right? So that means what is in the juice X? Basically, juice X contains the disaccharide called sucrose. Sugar cane contains sucrose. Okay. Now, can the sucrose diffuse out of the Visking tubing? It cannot. Can the iodine diffuse into the Visking tubing? Can. But can the iodine react with the sucrose? No. Iodine is used to test for the presence of starch. It does not react with the sucrose, okay, in the Visking tubing. So there's no change of color in the Visking tubing after 15 minutes, okay? No change of color. And the explanation is that the juice X here contains sucrose, a disaccharide, right? And the iodine does not react with disaccharide. It only reacts with the starch, which is a polysaccharide. All right. So 1 mark for your prediction, no change of color in the Visking tubing after 15 minutes, and 1 mark each for any one of these P2, P1, P2, and P3 answers. 2 marks, sorry, because total marks are 3 marks. Okay, so this is what you can write inside. Now, other important questions. So here we are going to discuss questions related to variables, hypotheses, and operational definition. All right, based on this experiment. Okay, first question, F for variables. State the variables for this experiment. What is the manipulated variable? It's the size of the molecules, right? And then what is the responding variable? Now, the presence of molecules in the Visking tubing and beaker after 15 minutes, or the change in color, the final color, change in color or final color of the solution in the Visking tubing and in the beaker after 15 minutes. Okay, textbook uses one, presence of molecules in Visking tubing and beaker after 15 minutes. Okay, then the fixed variable according to textbook is the surrounding temperature. Now, hypothesis. G, state the hypothesis for this experiment. So based on textbook, only small molecules are able to diffuse through a selectively permeable membrane, while large molecules cannot diffuse through a selectively permeable membrane. Okay, and the small molecule here is the iodide ions or the iodine molecules, and the large molecule is the starch. Now here's another prediction question, a different one from the previous one we have discussed. A similar experiment is carried out, but this time solution P is replaced with grape juice. Okay, predict the outcome at the end of the experiment when the solution in the beaker is tested with Benedict solution. So underline this word, grape juice. What is the outcome at the end of the experiment? When the solution is tested with what? Benedict solution, not iodine, Benedict solution. So what is the content of grape juice? What does it contain? Yes, grape juice contains glucose molecules. So imagine the glucose molecules are in the Visking tubing. Okay, solution P is in the Visking tubing. So the glucose molecules are in the Visking tubing. After 15 minutes, right, what happened to the glucose molecules? They are small enough because glucose is a monosaccharide. The molecule is small enough to diffuse out into the solution in the beaker. Okay, then you test that solution in the beaker with Benedict solution by taking a sample of the solution in the beaker and then mixing with 2 ml of the solution in the beaker with 1 ml of Benedict solution or 2 ml of Benedict solution. Then you heat it. You will get a brick red precipitate. So prediction is a brick red precipitate is formed when the solution is heated with Benedict solution. Explanation. Okay, 1 mark for your prediction and 2 marks for your explanation. Grape juice contains glucose molecules, which are small in size, right? And these glucose molecules are able to diffuse through the Visking tubing into the solution in the beaker. So when you test the solution in the beaker, you will get a brick red precipitate because glucose is a reducing sugar that produces a brick red precipitate with Benedict solution, or it's a reducing sugar that reduces the copper 2 sulfate in Benedict solution, right? So you get the brick red precipitate. So this kind of question can be asked in the exam too. All right. Now, lastly, operational definition. Okay, so let's revise how to write an operational definition. Based on the experimental result, explain the operational definition of diffusion. All right, so the tip here is you must remember the components of an operational definition as I've discussed with you in previous videos. Know the basic definition of diffusion first. All right, and secondly, adapt it to include the organism, solutions, and materials used. All right, so next, this is 1 mark for it. Then the second point is you must write, it is determined or shown by, the diffusion is determined or shown by the sub RV. So know your sub RV and write it completely, okay, in this answer. And then RAM, the third one is RAM, okay, R for RV, A for affected by, and M for MV. So RV is affected by MV. Know your MV too in this experiment, which is actually the main and sub MV is the same here, right? Which is the size of the molecules, right? So okay, try and write your answer first, and then you're going to correct it. When you correct your own answers, you are like the teacher, and you will know then what your weakness is and how to correct it. Okay, so it's good to mark your own paper. Right, let's look at the answer. Firstly, start with the term that you are going to define. Diffusion is, or whatever the term is. Okay, so diffusion is the movement of molecules P and Q across a Visking tubing down the concentration gradient. Now, if you look at the textbook definition of diffusion, it says that diffusion is the movement of molecules from a region of higher concentration or an area of higher concentration to an area of lower concentration, right, down the concentration gradient. So we adapt it. Now, diffusion is the movement of molecules, but here your molecules are P and Q, so you mention P and Q. Don't mention iodine or starch molecules. Okay, it's molecules P and Q according to your experiment. And you use a Visking tubing. Okay, so you must stay across a Visking tubing. Actually, diffusion sometimes doesn't need a semi-permeable membrane, but in this case there is. Okay, it's the Visking tubing. Down the concentration gradient. That means from region of high concentration to region of low concentration. Okay, and then the second point is shown or determined by the final color or color change of the solutions in the Visking tubing and beaker after 15 minutes. So this part here, the final color or color change of the solutions in the Visking tubing and beaker after 15 minutes, this is your sub-RV. So you must write is shown or determined by the sub-RV. Lastly, is affected by the size of the molecules. That is your MV. So any two of these points, you get 2 marks, right? So it's not that difficult. So remember the acronym DOREM. D for that basic definition, O for organisms, solutions, materials, R for is shown by the RV, sub-RV in the experiment. Okay, not the main RV, the sub-RV. And then RAM is for RV affected by MV. Right, that's all for this lesson. I hope that you have benefited from this revision and continue to study, stay calm and study, all right? And stay positive. And thanks for viewing all these, viewing the videos. Please do share, like, and comment also in the below in the comment section, right? So that's all for this lesson, and wish you all the best in the coming exams, especially the SPM. Goodbye for now.